Hello guys, welcome to my channel. So as you know, we are studying about the enzymes. So if you saw the last video, we just did the introduction for the enzyme. Now we'll go much deeper in the enzyme, like how enzyme works. So the enzymatic catalysis of reaction is essential to living systems, correct? And understanding of, or like under any biologically relevant conditions, uncatalyzed reaction tend to be slow, and most biological molecules are quite stable in neutral pH, mild temperature, aqueous environment inside the cell, right? So reaction required to digest food, send nerve signals, or contract muscles simply do not occur at a useful rate without catalysis. So that's why catalysis is very important. Right, so the distinguishing feature of an enzyme catalyst reaction is that it takes place within the confines of a pocket of the enzyme on the enzyme called active site. So for example, like just a rough diagram, like this is an enzyme for example. So like in the enzyme, like in the center of its pocket, let me use different color. For example, this is the center, and here, like I will just make a dot. So in the center, the feature is like the enzyme catalyzed reaction takes place within the confined pocket of an enzyme. So it's like a confined pocket, and like for example, it takes here. So this is known as an active site, right? And the molecule, so the molecule that is bound in the active site and acted upon by an enzyme. So for example, here is a molecule, like a star-shaped molecule, for example, that is reacting with here with the enzyme. So the molecule that is bound to the active site, and this is acted upon by an enzyme. So this molecule is called substrate, right? So the center part where the enzyme catalytic reaction occurs, that is called an active site, and the molecule that is acted upon by, that is called a substrate. Right, and the surface of the active site is lined with amino acid residues, right, with substituent groups that bind the substrate and catalyze its chemical transformation. Or often the active site encloses a substrate, sequestering it completely from solution. Alright, a simple enzymatic reaction might be written as, alright, so I will just remove this. So simple enzymatic reaction is like this, for example, E plus S, so that is both sides, correct? That might be ES, this is again both sides, that is EP, and that is E plus P product, correct? So this is just a simple enzymatic reaction where E is the enzyme, right? S is the substrate, P is the, that's a P, that's a product, right? And ES and the EP are the transient complexes of the enzyme with the substrate and with the product. So to understand catalysis, we must first appreciate the important distinction between reaction equilibria and a reaction rate. So what's the difference between reaction equilibria and reaction rate? Or you can say an equilibrium and a reaction rate. So the function of catalyst, as we know, is to increase the rate of reaction, right? I'll write down here, function of catalyst is to increase the rate of reaction, right? All right, so I will draw a diagram. All right, so for example, this is this, right? This is P. That's the product. So I'll explain everything to you. That's the transition state, right? And here, that's there. Okay. All right. So just out here. Ground state. All right. All right. So for this diagram, so as we know, the catalysts do not affect the reaction equilibria or the equilibrium. It does not. It just affect the rate of a reaction. So this diagram shows this is a substrate and that's the product. So it just shows S to P and P to S or the either way. And this picture also shows the energy changes during the reaction, right? So the delta G that you can see, so that's a free energy. That's the symbol of free energy, right? So in this diagram, the free energy of the system is plotted against the progress of the reaction. So the starting point, so this is the starting point. So starting point for either the forward, so this is the forward, or either the reverse reaction is called the ground state. So, right, that's the ground state for if it goes to product to substrate, that's the ground state. And if it goes to substrate to product, that's the ground state. All right, and the contribution to the free energy of the system by an average molecule under a given set of condition, right? So that is shown here. Okay, so the equilibrium between the S and the P, that's the product, reflects the difference in the free energies of that ground state. So in this example, the free energy of the ground state of P, right? The P is lower. So P is lower than that of S, right? That's the difference. So the S energy is here, right? And the P is here. So the free energy of the ground state of the product is lower than that of the substrate. So that means the delta G or the free energy for the reaction is negative, right? So the reaction is negative and that's why the reaction is exergonic, right? Because it's negative. If it's positive, like if, for example, this P is above here at this point, so the free energy of the reaction will be positive and the reaction will be endergonic, right? And so because it is negative and at the equilibrium, so at the equilibrium, more product than the substrate, right? The equilibrium favors the product. So the position and the direction of equilibrium are not affected by any catalyst, right? So the position and the direction are not affected by any catalyst. All right, so a favorable equilibrium does not mean that the S to P conversion will occur at a detectable rate. So the rate of reaction is dependent on an entirely different parameter. So like what parameter? So there is an energy barrier between the S to P. So the energy required for alignment of reacting groups or for formation of transient unstable charges or bond rearrangements and other transformation required for the reaction to proceed in either direction. So these are the things that is required if it goes on this way or if it goes on that way, right? So like alignment of reacting groups, formation of transient unstable charges, bond rearrangement, this all things comes when it goes either way, right? And so that's why there is an energy barrier and this energy is required to do all this different functions, right? So to undergo the reaction, the molecules must overcome this barrier, right? And therefore must be raised to a higher energy level, right? As if it wants to go to S to P, it has to create a higher energy level to go from S to P. So this is at the top of the energy hill that you can see here, right? Is a point at which the decay to the S or the P is or P state is equally probable, right? It's a downhill on both sides, right? So that's why this is called transition state. That's why I wrote transition state. Here you can see, and now you can understand. So at the top of the hill or the energy hill is a point at which the decay to the S or to the P state is equally probable. Both sides equally probable. This is called transition state. So the transition state is not any chemical species with any significant stability and should not be confused with the reaction intermediate. That is this AS or EP, right? It is not like enzyme and substrate. It's here, or enzyme and product is here. It should not be mixed. Reaction intermediates are different. Alright, but this is just a transition state. Right, so it is a simply, what is a transition state? So it is simply a fleeting molecular movement in which events such as bond breakage or bond formation and charge development have proceeded to the precise point at which the decay to the substrate or decay to the product are equally likely, correct? So that is the transition state. So when it reaches a transition state, right? So the events such as bond breakage or bond formation or charge development have proceeded to this point, and from that point the decay to the substrate and decay to the product are equally likely. So the difference between the energy level of the ground state and the transition state is the activation energy, right? So this we can say as a activation energy. So difference between the ground state to the transition state, right? That is the activation energy. So the rate of a reaction reflects this activation energy, right? So a higher activation energy corresponds to a slower reaction, right? If it's higher, then it goes to a slow reaction. So the reaction rates can be increases by raising the temperature or pressure, thereby increasing the number of molecules with sufficient energy to overcome the energy barrier. Right? So the reaction rate can be increased by raising the temperature or the pressure or increasing the number of molecules. Alternatively, or else the activation energy can be lowered by adding a catalyst. So catalyst enhance reaction rate, lowering activation energy, right? So catalyst enhance reaction rates, lowering activation energies, right? So this is what catalyst does. All right. So the bidirectional arrows you can see in this equation. So right. So any enzyme that catalyzes the reaction S to P or also P to S, right? Also catalyzes the same reaction, right? So the enzyme that is catalyzing S to P, that same enzyme catalyzed P to S as well. So the role of enzyme is to accelerate the interconversion of S and P, right? So the enzyme is not used up in the process and the equilibrium point is unaffected. But however, the reaction reaches equilibrium much faster when appropriate enzyme is present because the rate of reaction is increased. All right, so we'll see one more reaction. So this is all right. So this is a reaction that is a conversion of sucrose and oxygen to carbon dioxide and water. So writing down here, sucrose and oxygen to dioxide and water. Right, so this conversion takes place through a series of separate reactions and has a very large negative delta G, or the free energy, and at equilibrium the amount of sucrose present is negligible, right? So yet sucrose is a stable compound, right? Because the activation energy barrier that must be overcome before the sucrose reacts with the oxygen is quite high, right? So the energy required, that is quite high. So sucrose can be stored in a container with oxygen almost indefinitely without reacting. So it won't react. But in cells, however, sucrose is readily broken down to carbon dioxide and water in a series of reaction catalyzed by enzyme, that is catalyzed by enzyme. So these enzymes not only accelerate the reaction, but they organize and control them so that much of energy that is released is recovered in other chemical forms and made available to the cell for other tasks, right? So this is how enzyme works. So the reaction pathway by which sucrose and other sugars is broken down is the primary energy yielding pathway for cells, right? And the enzymes of this pathway allow the reaction sequence to proceed on a biological useful time scale, right? So any reaction may have several steps involving the formation and decay of transient chemical species called reaction intermediates, right? So what is a reaction intermediate? Is any species on the reaction pathway, for example, there's a reaction pathway that has a finite chemical lifetime, right? So for example, when S is converting to P or P to S, reaction is catalyzed by an enzyme, right? ES and EP complexes can be considered. These are the intermediates, even though S and P are the stable chemical species. So ES and EP complexes occupy valleys in the reaction coordinate. Right, so what does that mean? Valley, it's like here. ES and EP, right? So the complexes occupy this valley in the reaction coordinate diagram. So additional less stable chemical intermediates often exist in the course of an enzyme catalyzed reaction. So the interconversion of two sequential reactions intermediate thus constitute a reaction step. So that constitute a reaction step. So when several steps occur in a reaction, the overall rate is determined by the step or steps with the highest activation energy, and that is called a rate limiting step. So in a simple case, the rate limiting step is the highest energy point, right? This is the highest energy point for interconversion of S and P. In practice, the rate limiting step can vary with reaction condition, and for many enzymes, several steps may be similar activation energies, which means they are all partially rate limiting. Right, so the activation energies are the energy barriers to chemical reaction, and this barrier are crucial to life itself. Why? Because the rate at which a molecule undergoes a particular reaction decreases as the activation and barrier for that reaction increases, correct? So without such energy barriers, complex macromolecules would revert spontaneously to much simpler molecular forms, and the complex and highly ordered structure and metabolic processes of cell could not exist, right? So over the course of evaluation, enzymes have developed to a lower activation energies selectively for reaction that are needed for cell survival. All right, so the enzymes are extraordinary catalysts, or enzymes are also very specific as well, readily discriminating between the substrates with quite similar structures. So how can this enormous and highly selective rate enhancement be explained, or what is the source of the energy for the dramatic lowering of the activation energy for specific reaction? So all these answers has two distinct or but two interwoven parts. The first is in the rearrangement of covalent bonds during an enzyme catalyzed reaction. So the chemical reaction of many types takes place between the substrate and enzyme, right? Or a functional group, specific amino acid side chains or metal ions and coenzymes. So the catalytic functional group on an enzyme may form a transient covalent bond with the substrate and activate it for a reaction. Or a group may be transiently transferred from the substrate to the enzyme, right? So this case where it can be linked with the covalent bond and it can be activated, right? So the covalent interaction between the enzyme and the substrate lowers the activation energy and thereby accelerate the reaction by providing an alternative lower energy reaction pathway, right? The second part of the explanation is in the non-covalent interaction between the enzyme and the substrate, right? If you remember, like when we discussed about non-covalent bonds, so that are weak non-covalent interaction that help stabilizes protein structure and protein to protein interactions. So this same interaction are critical to the formation of complexes between protein and small molecules, including enzyme substrate. So much of the energy required to lower the activation energy is derived from the weak non-covalent interaction between substrate and enzyme. So what really sets enzyme apart from most other catalysts is the formation of specific ES enzyme substrate complex. So the interaction between the substrate and the enzyme in this complex is mediated by the same forces that stabilizes protein structure, including hydrogen bonds, ionic interaction, and hydrophobic effect, right? So the formation of each weak interaction in this enzyme substrate complex is accompanied by the release of small amount of free energy that stabilizes the interaction. And the energy derived from the enzyme substrate interaction is called a binding energy, right? So then the energy that is derived from the enzyme substrate interaction, that is called a binding energy, right? And its significance extends beyond a simple stabilization of enzyme substrate interaction, right? So the binding energy is a major source of free energy used by enzymes to lower the activation energies of reactions, right? So two fundamental and interrelated principle provided a general explanation for how enzyme use non-covalent binding energy. So much of the catalytic power of an enzyme is ultimately derived from the free energy released in forming many weak bonds and interaction between an enzyme and its substrate, right? So this binding energy contributes to specificity as well as to catalysis, correct? And weak interaction are optimized in the reaction transition state. So enzyme active sites are complementary not to substrate per se, but to the transition state through which substrate pass as they are converted to product during an enzymatic reaction, right? So this is the base to understand the enzymes, and that's why it was our primary focus, right? So how enzyme and substrate come together and how the non-covalent bond and the covalent bond that is providing free energy to lower the activation energy of the reaction. Right, guys, so that was the second part for the enzyme. We'll go through the enzyme. It's a long topic. All right, thank you guys for watching and we'll see you in the next video. Thank you.